it's time to turn to our next guest speaker. So from University of Alabama in the United States, graduate students and member of the BIP Lab, Allan Umfress has taken part in the University of Alabama training program in the neurobiology of cognition and cognitive disorders. The CCND program provides a solid foundation in molecular, cellular, and cognitive neuroscience theories and findings, as well as training in the mechanisms for translating basic research into advancements in the treatment of cognitive disorders. Allan Umfress is one of the authors of papers such as neuropathological effects of chemotherapeutic drugs, or characterization and preclinical treatment of rotational forced induced brain injury, as well as a researcher implicated in the developing of a new drug that could help stop depression, anxiety, brain injury, and cognitive disorders. The preclinical drugs works by inhibiting the cyclin-dependent kinase 5, which is found in mature neurons. Now I want to introduce you to the topic. The name of the topic is systemic administration of a brain-permeable cyclin-dependent kinase 5 inhibitor that alters neurobehaviors. Dear participants, let's now welcome Allan Umfress.
Wow, thank you so much. That was such a nice introduction. Hold on, let me see if I can share my screen and then get my talk up. Maybe this one. Hold on, give me 1 second.
No worries, take your time.
Yeah, I'm working on it. But that was very nice. So today I'm going to talk about the CDK5 inhibitor, as well as some of the brain injury modeling that we're doing. You guys see that?
Yeah, perfect.
Wonderful. All right. So kind of as it was just alluded to, we focus on different forms of neuronal injury. So like one is a traumatic brain injury. And another one that we focus on is this concept called chemobrain, in which you have off-target effects of chemotherapies. And we focus on therapeutic strategies to sort of prevent these injuries. And we do this in preclinical rodent models. So, you know, very distinct from the last talk you just have, but neuroscience is sort of a broad, expansive field. And I really love that about this conference, because we can kind of appreciate that. So what we do in our lab is we take the big brain and how it functions in cognition, and we focus on these, you know, billions of cells within the brain, these trillions of connections that are formed on each of these cells, which have these things called synapses. We have a presynapse and a postsynapse. And we focus even more specifically about the signaling molecules within these synapses and how they communicate, how they become aberrant or disoriented in a disease-like state, and then how we can target these to treat the diseases. So it's a very reductionist approach to neuroscience. And the way we model this is through traumatic brain injury. So traumatic brain injury is sort of any insult that happens to the brain that ultimately impairs your cognitive function. So this, you know, happens in epidemic proportions worldwide. Here I'm just, you know, showing some stats from the United States. But, you know, just in the United States, about 1.7 million people sustain a TBI. This is a leading cause of death, as about, you know, 52,000 of these people die every year. You know, hundreds of thousands are hospitalized. And it's sort of a silent epidemic, because most people who sustain a brain injury, about 80% of those people, they have their injury, they go to the emergency room, they're immediately treated based off of, you know, like basic cognitive tests and motor function and pupil dilation, and then they're sort of released from the emergency room. So immediately after you have these sort of brain injuries, you have these acute symptoms that are pretty obvious. If you guys have ever had a head injury, you know, most people have a headache or some sort of sensitivity to light and sound. You feel fatigued, dizzy. Ultimately, this sort of affects your behavioral functions, such as, you know, problems with your memory or sort of anxiety. And then as the pathology of a brain injury sort of progresses throughout the lifetime of a survivor, you have more long-term effects of brain injury. And these include increased risk for dementia, increased risk for depression, behavioral abnormalities such as social isolation, aggression, psychosis, and long-term vision issues. So the reason I said it's really a disservice to patients to treat them immediately in the ER and then release them is because the pathology of a brain injury is very expansive, much longer than the hour from your ER visit. So initially, when you sustain a brain injury, you have compression of the brain tissue, and this induces, cellular death, tearing of the axons. You have damage to the vasculature, which causes hemorrhages throughout the brain. So you have blood leaking into your brain, essentially. And this occurs, rapidly after the head is impacted by something. So this is in seconds to minutes after your injury. But then you also have this secondary damage that occurs in hours and days after your injury. So this includes, secondary waves of apoptosis or cellular death, inflammation that continues to progress in different cortical areas of the brain, demyelination, and then different pockets of secondary ischemia. So where you've had damage to the vasculature in the brain, and now the nutrients that are being carried by your blood are no longer able to get to certain areas of your brain. Okay. And then, in the months and years that follow a brain injury, you actually have neurodegenerative phenotypes. You have substantial loss of neurons. You have what's called tau neurofibrillary tangles. So this is something you see in an Alzheimer's disease or any other neurodegenerative condition. So those people who sustain a head injury actually have a 2.3-fold increased risk of developing Alzheimer's disease. You have a 1.5 increased risk of developing Parkinson's disease. And here you can just see an image of the ventricles of one patient who suffered a brain injury. Within 2 years, you see this rapid expansion of the ventricles, suggesting, substantial loss of brain tissue over time. So as I'm mentioning, it's a disservice to patients to treat them immediately and then release them because they seem fine, because the damage from a brain injury is progressive and it's occurring throughout the lifetime of the survivor. So, and then as I mentioned, this leads to, long-term negative consequences such as, Alzheimer's disease, and then also brain injury itself. Repeated brain injury leads to its own neurodegenerative condition, and this is a condition that we call chronic traumatic encephalopathy. So here you can see substantial degeneration of the brain from a patient who's had CTE. And this is characterized by increases in neurofibrillary tangles, which is very synonymous to like an Alzheimer's disease. And this is really popularized in the last couple of years. So in the States, we play like American football. And it was really famous because they looked at 111 brains of former professional football players. And out of 111, they noticed that 110 of them had these degenerative phenotypes, suggesting that head injury itself induces a unique form of neurodegeneration. This is deemed chronic traumatic encephalopathy. So as I mentioned, we focus on different ways to test brain injuries and therapeutics preclinically. So preclinically, we model this in mice and rats. And there's really only a few ways to model a brain injury in a mouse or a rat. So right here, on the left, we have what's called a weight drop model. So that's pretty self-explanatory, right? The animal is restrained and you drop a weight on its head to mimic a brain injury. Here in the middle is an example of what's called a controlled cortical impact. So this is where you perform a craniotomy on the rodent and you insert an impactor into the brain to compress and damage the brain tissue. And then another more popular model is this model. It's called a blast model. So this is where a percussive sound wave is sort of explodes a mylar sheath next to the animal's head. And this energy wave sort of vibrates the brain within the skull. So this is supposed to mimic like a explosion in like a war zone, because veterans of wars often suffer traumatic brain injuries. And while all of those are good models in that they recapitulate different aspects of brain injury, like cellular death, damaged vasculature, injuries to the cortex, when you look at how most patients present to the clinic, most head injuries are caused by a rotational acceleration and deceleration of the brain. So most brain injuries result from falling, a road traffic accident, a sports-related event, and all of these injuries are injuries in which the brain is very rapidly rotating, accelerating and decelerating in the brain. And this causes shearing forces that tear and damage the axons within the brain. And as I sort of mentioned before, all of these models are more linear-based models that are just compressing the top cortex of the brain instead of rotating the brain and damaging subcortical axons. So, yeah, as I alluded to, the rotational injuries are what induces the shearing forces that ultimately tear the axons, so that the ways our neurons are communicating with each other. So, The first part of our project was to design a new model of brain injury, one that would be more rotational and would mimic sort of this clinically relevant form of brain injury. So we worked with a team of engineers and we designed this rotational brain injury model. Essentially, it's a large pendulum with a swinging arm. The animal is fit into a restrainer at the base of the pendulum. The pendulum is equipped with an accelerometer, so this is an inertial measurement unit device. And it swings very rapidly and it comes in contact with this ventral strike plate at the bottom of the base of the pendulum. And then it rotates the animal's head in 90 degrees both ways to make a clinically relevant model of brain injury. Here, I have a little video to show how this would work. Right, so the animal is propelled forward very rapidly. It comes in contact with this strike plate at the base, and the head rotates very rapidly both directions to mimic a rotational brain injury. So following rotational brain injury, we wanted to see that this could mimic some of the pathologies we see in the human condition. And one of the pathologies that's most relevant in human conditions is a prolonged neuroinflammation. So one way we monitored neuroinflammation in these animals was by using PET imaging. So we use PET imaging for this radioligand. It's called TSPO. So TSPO is a translocator protein that's very lowly expressed in the central nervous system. However, when microglia and macrophages, so the cells that are inducing inflammation, whenever those become activated, this marker becomes heavily upregulated. So we can use this as a tracer for neuroinflammation within the brain. And we can see that following a rotational head injury, animals sustain increased inflammation throughout many areas of the brain, such as the amygdala, the CA1 region of the hippocampus, and the basolateral amygdaloid nucleus. And then we can go in and look at these brains histologically and verify the things that we're seeing with this non-invasive imaging technique are occurring histologically. So here we can see a stain for IBA1 positive microglia and GFAP positive astrocytes. And we can see that both of these markers of inflammation are increasing in the brain in the acute phase after injury. And then ultimately, when we induce a TBI to these animals and then we let them age for 6 months, we can stain for these tau tangles, so these neurofibrillary tangles that are associated with CTE and Alzheimer's disease. And we can see that in the brains of these TBI animals, they show increases in these neurofibrillary tangles. So this is all suggesting that by modeling a rotational injury in a rodent, we can mimic some of the pathologies that we see clinically and in some of these areas that are more relevant than previous models. So some of these subcortical areas. And this induces, similar pathologies that we see in the acute phase as well as the chronic phase. So then, the next thing we wanted to look at was the memory function of these animals, so their cognitive abilities, because a large problem reported by patients who suffer a TBI is impairments in retrograde and interrograde amnesia. So one way we can test memory function in rodents is by this paradigm called fear conditioning. So essentially, you place an animal in a box and it's got an electrically gridded floor. You play a tone and then the animal receives a mild shock. And then 24 hours later, you put it back in that same context. And you can measure the animal's freezing responses because the animal has a memory of this adverse event that happened yesterday. So it's going to freeze in response to this. And this is called contextual memory. So this is based off of hippocampal circuits within the brain. And then vice versa, you can place the animal in a novel context, so a context it's never seen before, and you can play the tone that it heard before it received the shock, and this is a memory of that cue. So it knows that this is a negative cue, that means an adverse event is going to happen, and you can measure its freezing response there. So this is more of a amygdalary circuit-dependent memory. So when we look at these memory functions in our animals that have sustained a TBI, we see that at baseline there's no alteration in memory. Right, so before we shock them, but then 24 hours later, after we've trained the animals, we can see that they have a significant reduction in their freezing behaviors, both in the contextual memory paradigm and in the cued memory paradigm. So this is suggestive that they have some sort of memory impairment following their injury, and this is independent of any sort of pain sensitivity following a brain injury, as they have the same responsiveness to adverse shocks in their abilities to flinch, to jump, or to vocalize their pain in response to a shock. So now that we know that we have this sort of mnemonic deficit within these animals, we wanted to look at the circuitry that's involved. So as I mentioned, one area of the brain that's responsible for your contextual memories is your hippocampus. So because we utilize rodent models, we can actually dissect out the brains of these rodents, and we can cut their brains into thin slices. And then we can stimulate and record from the electrical circuits of these brains in an artificial cerebral spinal fluid, setup. So here is an acute brain slice where you can see the hippocampus. And then here I'm showing you what we're going to do is we're placing a stimulating electrode here, and we're going to record from the axons that project to here. And we're going to look at how this sort of neurophysiological function is impaired following a brain injury. And when we look at this, we can see that, there's no alteration in baseline synaptic excitability. So that the circuits have the ability to function much like control circuits. So this is called an input-output curve. A paired pulse ratio is a metric of short-term facilitation that we look at to measure synaptic output. But really, the most important thing is that much like when you learn something or when you have a memory, you have a synaptic strengthening of those circuits responsible for that memory. So when we induce what's called long-term potentiation, so this is sort of the cellular correlate of memory is what people like to say. By inducing a high frequency stimulation to these circuits, you see a robust increase in this circuit amplitude in control animals. And this is significantly depleted in TBI animals. So they're unable to induce a long-term potentiation of their synapses, which ultimately leads to memory impairments in these animals. Okay. So as it was sort of mentioned in the introduction, and then we wanted to study the different biochemical mechanisms that are occurring within these animals. And one way we do that is by focusing on a particular kinase, and this is called cyclin-dependent kinase 5, so CDK5. So CDK5 is sort of a unique cyclin-dependent kinase because it doesn't require cyclin to be activated at all. So its name is sort of a misnomer. Instead, it has a unique coactivator. This unique coactivator is this protein called P35. Okay, and CDK5 P35, this holoenzyme of these two complexes, has numerous functions within a nerve cell. So it's involved in axonal transport, it's involved in dopamine signaling, it's involved in receptor trafficking. However, under excitotoxic conditions, so any sort of condition in which calcium homeostasis is dysregulated, or cells are sick and cells are dying, so much like a brain injury, you have an increase in intracellular calcium, and this activates a protease. This protease is called calpain. So calpain cleaves this P35 protein right here at this site, and that creates two new proteins. So it creates this protein called P25 and this protein called P10. So P25, much like P35, is able to activate CDK5. But this is sort of a unique activation. We call it an aberrant activation because P35 has this meristylation. So this keeps it sort of bound to the membrane. And then also within these domains are the different regions with which P35 is degraded by the proteasome. So this is how we get rid of this protein in our cells. So now that we've lacked this meristylation and we lack the ability to degrade this protein, we now have an aberrant complex that is mislocalized. So it's no longer bound to the membrane and it's harder to degrade. So we call this an aberrant or hyperactive kinase. Okay. And this hyperactive kinase has been implicated in numerous neurodegenerative conditions. So there was this famous Nature paper in 1999 where they showed that this P25 protein is upregulated in the brains of Alzheimer's disease patients. And when you use genetic tricks, so this is using a tetracycline transactivator to overexpress this P25 protein, so it's just a genetic tool, you induce inflammation in the brain just by increasing the expression of this protein. And over about 6 weeks of overexpressing this protein, you see degeneration of the synapses, the spines, where these neurons are forming connections, and you see reductions in brain weight in mice. So all of these are suggesting that this P25 protein is a bad protein, and it's not something you want upregulated in your brain, but it's upregulated in the brains of Alzheimer's disease patients, and it's upregulated whenever you have something toxic happening to your brain. So we went on to show that this toxic signaling is occurring in different forms of brain injury. So this is an example of a controlled cortical impact in which you impact the mouse brain and you see cellular death. So here we're looking at pycnotic neurons or fluorojade positive degenerating neurons. But when we use a genetic tool, so this is a CRE tool to eliminate this CDK5 gene, and we give animals these head injuries, they're now neuroprotected from these injuries. So here you can see a wild type animal where it has this large necrotic core where all the tissue is dying. And here you can see a CCI in which we've knocked out the CDK5 gene. So once we knock out this one enzyme, these animals are neuroprotected from these injuries. So the injuries are much less severe and they have better cognitive function after their injuries. So there it goes. All right, so that's all nice and dandy, but we can't really knock out a gene in a human. So we wanted to develop pharmacological agents that we could use to treat these injuries. And we wanted to develop pharmacological agents that could target this CDK5 enzyme. So we worked with a medicinal chemist and we screened a bunch of CDK5 inhibitors, so things that would inhibit its function, and it's a kinase, so we can measure its function by the phosphorylation of its substrates. So by treating different brain tissues with these inhibitors, we could see that in ex vivo conditions, so when we take out a brain slice and we treat that brain slice with these drugs, we see reductions in CDK5 activity across all of these inhibitors that we tested. However, only one inhibitor, this inhibitor called 25106, which had about a 30-fold more specificity for CDK5 over CDK9, so a very similar kinase. Only one of these inhibitors was able to cross the blood-brain barrier and inhibit CDK5 function in vivo. So we saw this across a 24-hour time period and across 10 to 200 mg per kg of dosing the animals. And this mechanism of action had to do with its ability to bind the kinase's ATP binding domain. So it inhibits its ability to bind ATP, so it can no longer function as a kinase to add phosphatate groups to things. So then we wanted to determine, the kinetics of this drug within the brain. So to do that, we developed a mass spectrometry technique in which we look at the molecular weight of the parent compound versus a fractionation product. And then we can generate a standard curve of this using known concentrations. And then we can take samples of blood or brain from animals, and then we can determine how much of this drug is getting into different organ systems. And when we did that, we noticed that the drug was rapidly uptaken into the blood, the plasma, and into the brain of animals. But uniquely, it displayed linear kinetics within the plasma in that, the more drug you take, the more drug you have in your plasma. But within the brain, it showed exponential kinetics. So the more and more drug you take, the more and more is getting into the brain at an exponential rate. And it had decreases in brain clearance over the time and over the doses that we give them. So the more drug you give these animals, the more it's getting into their brain and the more ability it's going to have to inhibit this kinase, this CDK5. So all of this sort of suggested that this drug was a good therapeutic that we could deliver systemically to treat different forms of injury. So another thing we wanted to assess was, well, we knew that when we knock out this CDK5 enzyme, that the animals were neuroprotected from injury. And we also knew that they displayed distinct behavioral phenotypes. So one of those phenotypes was that they were very hyperactive. And one of those phenotypes was that they were very resilient to stress. And one way we model stress in mice is through a test called the tail suspension test. So mice are suspended by their tail and you measure how long they struggle for. So here's like an example of this, in which you can pick up a wild type animal, and they struggle a little bit, but ultimately they sort of give up. But when you knock out the CDK5 gene and you do the same thing, the animals are very hyperactive. They struggle a lot. They're very resilient to stress and different stress induction paradigms. So ultimately, when we give animals our inhibitor of this enzyme, much like knocking out the enzyme, we should see the same behavioral phenotype. So we can put the animals in an open field and we can track their behavior using a computer and measure how they move around. And we noticed that when we inject animals with a CDK5 inhibitor, much like the knockout phenotypes, We say that they're very hyperactive, so they run around they explore the open area of the mazes a lot more. They travel around a lot more. They're hopping and sniffing and rearing a lot. And then when you suspend them by their tails, much like the video I just showed you, they move around almost constantly the whole time. So they never go immobile, and they spend a significant amount of time, struggling and fighting this stressful condition, whereas control animals just tend to give up. Okay. So with this in hand, this new pharmacological agent in hand, and its ability to penetrate the blood-brain barrier, the next thing we wanted to do is say, all right, well, in different conditions in which we no longer have this CDK5-P35 complex doing its physiological role, but in context where we have CDK5-P25 doing its aberrant, cytotoxic role, can we then inhibit this enzyme and rescue different forms of injury? So one way we can test this in vitro models is by treating cells with high concentrations of NMDA. So NMDA activates NMDA receptors, and this increases your calcium. And this ultimately leads to pathological consequences within the brain. So here we can see that NMDA generates this P25 product, or when we deprive cells of oxygen and glucose, we get this same generation of this P25 product. And now we have this inhibitor that we can test in these cultures to see if that is going to neuroprotect in any way. So in our primary neuron cultures, we ultimately deprive these cells of oxygen and glucose. And we see that this reduces the cell viability by about 50%. So about 50% of these cells are dying. However, when we treat these cells with a CDK5 inhibitor, we get a rescue of about 25% viability. So this is suggesting that under instances of ischemia, so oxygen and glucose deprivation, this is neuroprotective. And then this is in cells, and then we can do the exact same treatment in slices where we're looking at, so TTC stain is a viability stain. So all the red areas here are cells that are alive, and all the white areas are cells that are dead. So we can look at this in the context of oxygen and glucose deprivation. We see a significant reduction in cell viability, so increased cell death. And then ultimately, this is neuroprotected when we treat with our CDK5 inhibitor. And then the same thing happens again when we do a different form of injury treatment, which is high concentrations of NMDA. And then this is ultimately neuroprotected when we use our CDK5 inhibitor. So back to our rotational brain injury model is we can see that, this is a model that induces, cytotoxicity by generating this P25 product and activating calpain proteases, which lead to the degradation of proteins. Now, can we treat with our inhibitor and rescue some of these negative effects of brain injury? So when we did that, initially we injured these animals, and then we immediately treated them with our CDK5 inhibitor. And then 7 days later, we tested their memory function in the fear conditioning paradigm I explained earlier. And we see that, these animals have a significant reduction in their freezing behavior. So they're having memory impairments. However, when we treat with this CDK5 inhibitor, this memory impairment is sort of rescued. And this is true in the contextual fear memory paradigm, as well as the acute fear memory paradigm. And then we will look at the pathological consequences of brain injury. So one of this is like neuroinflammation. We can see that TBI itself increases these neuroinflammatory cells. So these are astrocytes. However, when we treat with a CDK5 inhibitor, we see a reduction in these neuropathological consequences. And this is true when looking at neuroinflammatory astrocytes, as well as activated microglia. So these are IBA1 positive microglia. All of this together sort of suggests that this pharmacological agent and our knockout agents all together are really suggesting that this enzyme complex is neurotoxic. And by inhibiting this complex, we can rescue some of the negative consequences of brain injury in rodents. And this is nice, but we wanted to identify the different downstream mediators of the brain injury. So CDK5 is a kinase, and kinases act by adding phosphatate groups to their different substrates. And then this ultimately changes the functionality of these different substrates. So it can either activate them or repress them. So we can do a technique called phosphoproteomics. This is a look at the different landscape of phosphoprotein changes following any sort of treatment. So when we did this following brain injury, we can enrich for CDK5 phosphorylation motifs by using a CDK5 antibody. And we can identify hundreds of sites that are increased following brain injury and that are ultimately decreased when we treat with a CDK5 inhibitor. And, sort of remarkably, we saw that, of the 653 new phosphorylation sites that are occurring in brain injury, almost 72% of those sites were blocked when we treated with our inhibitor. So all of these are sort of the downstream substrates that are inducing cell death when CDK5 becomes aberrantly activated. And now we have a way to inhibit these and identify these. So then this is a pathway analysis looking at these different pathways and the different molecules that are involved. And then we can do the same thing with total proteomics. So not just looking at the phosphorylation states, but looking at how genomic changes are occurring in the brain injury and how inhibition of CDK5 alters these signaling cascades. So just kind of to conclude everything, we started with this ultimate idea that we needed a better preclinical model of brain injury, because ultimately, if we're not modeling the clinical condition, it's very hard to develop therapeutic approaches that we can translate to humans. And we did this by making a new model, by screening different CDK5 inhibitors. We identified this lead compound called 25106. We showed that using this compound, we could provide neuroprotection from brain injury. And then we used different, large data omic approaches, so phosphoproteomics, to identify, all the changes that are occurring within the cellular landscape. And then, I'd like to thank, everybody in the Bib Lab, our funding, and our collaborators. And then this is Birmingham, Alabama. This is where I'm from. If you guys ever want to come visit or you want to apply, you guys should all come to graduate school here. It's a great place. And I'll take any questions. Thanks.
Thank you so much. So we have a question in our Q&A. So were there any complications regarding other brain functions while using CDK5 inhibitors observed in mice?
So yeah, as I mentioned, when you knock out the gene, the animals are kind of hyperactive. So we see the same thing when we treat with the inhibitor. And then CDK5 inhibition is really only a therapeutic strategy in the acute phase of injury. So when you knock out this gene and you let the animals age for a while, ultimately the cells become hyperactive, is what we would say. And this induces like a seizure-like behavior. So ultimately there could be a negative consequence of long-term inhibition, but we haven't assessed that yet.
Okay, thank you. Are there any other questions from our participants?
I have one question.
Yeah.
Did the size of the mice or weight and the race could have an impact on the substance 25106?
So we dose them based off their weight, right? So all the animals are weighed and given the exact same amount of dose depending on that weight. So no, there's no consequences of the weight yet. But thank you.
Okay.
Any other questions?
Well, I have a question. What kind of other species animal could this study be replicated?
I'm sorry, could you ask that again?
On what kind of other species animal could this study be replicated?
What kind of other animals could we use?
Yeah.
Yeah, so these
Can
different brain injury
replicate.
models. Yeah, so these different brain injury models are used for rodents. This could be used for mice and rats, but ultimately it's not the best model system, because if you look at the human brain, I showed it in my very first slide, it's large and it has gyrencephics, it has like these sulci, but ultimately when you look at, I don't know if you've ever looked at the brain of a rodent, but they're smooth-brained animals, they're less encephalic. So ultimately the idea would be to start with these smaller animal models and work towards larger animal models that mimic the human condition a little bit better. But right now it's only adaptable to rodents.
Thank you so much.
Yeah.
I think we're done with our questions. I want to thank you for speaking to our event. Your insights and expertise are invaluable, and we are sure that our attendees will benefit greatly from the information presented. Thank you again for your time and participation for the first edition of our conference, and we wish you a great productive day.
Cool, yeah. Thank you guys for inviting me. This was really cool. And then if anybody has a question or any of the students, you guys can email me. I provided my email and everything.
Okay,
so
perfect. Thank you so much.